Well, hey guys, first Screencastify for AP Bio, so we're going to see how this goes. This is Shelton here, kind of feeling like Mr. Anderson posting it, but first time is probably not going to be as good. So here is what I want for those of you who need me to kind of walk you through concepts and my explanations. Probably what you guys should have out is you should have out your handy dandy notes. I'm going to go through the PowerPoint with you guys and kind of just go over like where we left off because we did have a little bit of this lecture and then kind of talk a little bit about some of the newer stuff too, which is actually kind of fun. So with that, you may want to hit pause, and then you can go ahead and grab your notes outline if you wanted to do that, or have your computer, and your book might actually help you too, so that way you can follow along with any pictures if you want to. So, all right, so here we go. I apologize. Feel free to mute me or pause me whenever you want, but we are going to really try to go over and understand our chapter 21 concepts. So let's go to where is actually the very first slide that we talked about, which really was understanding populations and why diversity is so important within a population and a gene pool. So when we do talk about populations, do remember that populations, this would be a good example here of a population where it is all the members of the same species that actually have the opportunity to interbreed. So in this, what you're seeing here is this 40-mile herd range down over here absolutely doesn't reproduce up here, but there is this overlap that can actually happen. But it's kind of just recognizing that populations that evolve, it's not a species that can evolve. So that's what we're looking at here in chapter 21, the evolution of populations. Now, what we already addressed in class is we addressed genetic variation and why genetic variation is so important. Now, really what I need you guys to be able to address for the College Board test is what are the sources of that variation? I think you already know that you have to have variation in order to be able to adapt to an ever-changing environment. You know that. But I need you guys to be able to explain in eukaryotes our sources of variation, especially when we talk about sexual selection, or not sexual selection, sorry, that's to be another topic to be talked about a little bit. But sexual reproduction, and remembering with sexual reproduction, just those three key areas where we do get that genetic variation. So one being during meiosis, that crossing over, also understanding then that we have independent assortment, and then you also have that random fertilization too, that all gives every offspring just absolutely new sets of genetic combinations. Remember in bacteria, when we talk about bacteria and their sources of genetic variation, that's where we talked about one, obviously mutation. Mutation happens in anything that has any type of genetic information, but we also talked about a process of conjugation, we talked about transduction, we also talked about transformation. Didn't use those three terms, but we talked about actually how viruses can insert genetic information, how there can be an exchange of plasmids, and also DNA. Bacteria will also, prokaryotes, when conditions are harsh, they'll often just try to uptake surrounding DNA too in order to just get new genetic combination. So that's in our bacteria. Remember too, in viruses, a lot of the source of variation in viruses happens to be not just in the genetic material replicating, but also in that reassembly process that actually happens within the cell too. So just be aware of all of these different sources of variation. So that's really what 21.1 was having you guys review, our sources of variation and understanding that part. Now, when we went to, oh, such a cool picture. Love this. So, because remember, this particular picture here, what this particular picture is trying to show you is that natural selection is going to act upon phenotype, which changes the genotype in a population. But don't think because phenotype is different that it's actually the genotype that's different. Here, these are just two different caterpillars, same genetic combination, but their different diet is actually giving them different phenotypes, which is kind of cool. So just kind of keep in mind though, when we do talk about natural selection always does act upon phenotypes, and it's that act upon phenotypes that ultimately changes the genotype. Now, the most important part of this chapter, Artie Weinberg. So when we do talk about Hardy-Weinberg, what Hardy-Weinberg is looking at is changes in allele frequencies or in genotype frequencies in a population over time. Now, when we look up here and we see P or we see Q, P is the frequency of the dominant allele, Q is the recessive allele, and what we have to remember is P plus Q always equals 1. So when we report our frequencies are always in decimals. It's the same as a percent or it's the same as a fraction, but P plus Q always has to equal 1. And so we always have to remember that aspect of our allele frequencies. Now, when we put those alleles from the gene pool together, what we can actually use and predict is how are those going to be coming together in our genotypes within the frequency. So if we actually just look at this as basic math, I mean, Hardy-Weinberg, and they gave us some great equation, but they're kind of just doing some of these little guess and check tables that you guys learned how to do in algebra a long time ago. And with this, if we recognize that if we know P and we know Q, really our frequency of our homozygous dominant individuals is just going to be a result of P squared. And then if we actually address then the number of homozygous recessive individuals, it's just going to be Q squared. And then P times Q, we've got that twice. If we actually kind of look at this Punnett square or guess and check table, as we'd call it, and that's why our frequency of heterozygotes is going to be 2PQ. We have to absolutely know those particular equations. You will on the College Board test have an equation sheet that tells you, but it's really just better to know it because it's not that difficult. P plus Q equals 1. P is the frequency of your dominant allele, Q is your frequency of your recessive allele, and then P squared plus 2PQ plus Q squared equals 1. Those are your genotype frequencies for the population as well, where P squared, homozygous dominant individuals, 2PQ is your frequency of heterozygotes, and your Q squared then is your homozygous recessive individuals. So remember that. And then in solving, I gave you guys an equation or a worksheet in order to do that. To calculate your Hardy-Weinberg problems, you absolutely will have Hardy-Weinberg problems to solve on the College Board test, hopefully on May 11th. So let's just keep that in mind. Now, so do those practice problems and also remember the steps I gave you for solving Hardy-Weinberg problems too. What are you always going to start with? You are always going to be starting with information about Q squared. They will always give you information about Q squared, your homozygous recessive individuals. And then from that, once you know Q squared, find Q by square rooting it. Once you know Q, you know P because P plus Q has to equal 1. And then after that, just plug and chug into any part of the equation that you need to use is how you solve it. But always, guaranteed, you'll be given information about Q squared. So follow that first. Now, another part about Hardy-Weinberg that we need to make sure that we understand is that what Hardy-Weinberg actually said is for this equation to work. Now, if we actually take a look here of these generations, so here, these are changing frequencies. So these would not be in Hardy-Weinberg equilibrium because they're changing. Now, what they did say is if allele frequencies and genotype frequencies are staying the same, there were 5 assumptions we have to make about that particular population. So in a multiple, or not multiple choice, but in free response, what you absolutely also need to know are 5 conditions for Hardy-Weinberg equilibrium for allele frequencies and genotype frequencies to be stable. Here they are, the big 5. Write these down. We don't have a slide for it, so make sure that we are understanding them. So first and foremost, they said large populations. So that is number 1 of the conditions of Hardy-Weinberg. Because in large populations, that's a lot of allele frequency to change, and there's a lot of choice, and usually they stay a lot more stable. What we're going to address in a little bit is the fact that in small populations, there's actually a lot of allele change that can happen that's not due to natural selection. It's actually just simply due to random chance. And random chance can actually now change the allele frequencies or genotype frequencies, not natural selection and definitely not choice, but that's in small populations. We'll address that soon. Now, what they also, second assumption of Hardy-Weinberg, no natural selection. Duh. Natural selection happens, we're going to see that change in allele frequency. Now, what they also said is no mutation, mutation being that source of genetic change. So there's going to be sources of new introduction of alleles. Now, they also said too that there'd be no gene flow or immigration or emigration. So that's introducing new alleles or a higher combination of alleles into a gene pool. So that can also be a source of change. And then they also said that all mating was random in order to get that mix. And we're going to talk a little bit when we move into 21.3 about some of those differences. So 5 conditions of Hardy-Weinberg: large populations, no natural selection, no mutation, random mating, and no gene flow. So remember those 5, because anytime any of those 5 things aren't happening, we're probably going to be having some type of evolution of the population. Or, as we say in population genetics, there's going to be a shift in the allele frequencies of the gene pool. So let's move on to 21.3. So in 21.3, so let's just take a look at our notes outline, where we're at here in order to get down to it. So we just went over our 5 conditions of Hardy-Weinberg. Say them with me if you want to. I know I'm such a dork, but oh well. You miss me and you know it. Now, here's what we're going to be talking about in 21.3. In 21.3, we're going to just, it reviews again natural selection and this process of adaptive evolution. When we talk about adaptations, adaptations are these sets of phenotypes that allow an organism to be better adapted to its environment. And so that's really, when we talk about natural selection, adaptive evolution, really all kind of the same thing. Now, let's talk though about genetic drift, founder effect, bottleneck effect. Which of the Hardy-Weinberg principles is this addressing? Anyone? Anyone? It is addressing small populations. If I had a small population, these populations are subject to change just due to random events, not due to natural selection, not because they are characteristics that are better suited for the environment. Small populations are subject to these random events that lead to change. Now, 2 different ways of looking at that. That's what genetic drift is, these small populations that are subject to change. Founder effect, bottleneck effect, really just looking at the exact same situation, but in different scenarios. If I was going to talk to you about the founder effect, an example of the founder effect would be a group of finches blows over to the Galapagos Islands. Well, now that small founding group, that's your gene pool. Those are all the genes and the genetic diversity that founding population has to work with. Maybe that set of genes is not best suited, but they make do. So natural selection makes do with what's there. Now, another one is if we talk about a bottleneck effect. In a bottleneck effect now too, again, kind of have this random event that wipes out a large percentage of the population, wasn't natural selection, just random. And now you can see actually in this particular case that they lost whatever this yellow variation was just by random chance, not again by any natural selection action. Again, small populations, there can be some random events that lead to genetic change within that population. That's what we have to understand. Now, the other term that is in 21.3 is gene flow. And really, if we're talking about gene flow, we are talking about immigration, emigration, the movement of genes out or the movement of genes in is what we're addressing. So 21.4 is the next section that we need to talk about. And 21.4 is actually kind of fun stuff in here to talk about. Now, first term that we need to address in 21.4 is we need to understand what it means, that what relative fitness of an organism is. And when we talk about the fitness of an organism, so here's a greater prairie chicken that was listed there. But reality is when we talk about fitness in a biological sense, fitness really is, and the fittest organisms are those that contribute more to their population. And when I say contribute more, I don't mean lift more food. What I mean is they're contributing more of their genes to the population for future generations. So basically, if we're going to be measuring fitness of an organism, we're going to look at how many viable offspring do they produce. And do those offspring then get to reproductive stage? So that's what we look at when we're studying the fitness and even the relative fitness of an organism is their productivity in offspring. Now, when we talk about natural selection, one of the things, these graphs will be on your AP test. absolutely guaranteed you'll see it at least once or twice. But it's understanding that natural selection just doesn't go directionally one way or the other. It can when that actually happens, like here, if this is my original population and they're showing the diversity within the population here, in this particular scenario, what I have is few white ones, few gray ones, and a lot in the middle. Now, what can happen then is in this particular population, it favors with this directional selection, it favors the darker color. The light ones are the ones that are getting eaten by mice and birds. And so now this population is actually shifting, and the curve is shifting one direction or the other. So again, over here, this is my original population, and then this is the way it's shifting now in order to favor it. So it's going one direction, directional selection. Thank you when we actually have terms that make a ton of sense. Now, in this particular scenario here, in disruptive selection, well, what's actually happening here is this particular environment actually favors the light color, favors the dark color, but actually the unfavorable type is the middle or the average. And so in this, when it just gets chopped down the middle and now you've got two peaks, well, this is a disruptive selection. So we're disrupting that curve and creating two different curves. Now, this could also lead to a speciation event. That's next chapter. We'll talk about that then. And then the last curve is now the average is favored. The extremes, however, are not. So the extremes are getting selected out of the population, and what ends up surviving and reproducing more is more of this average particular color. So we get this narrowing of that particular band. So those would be our different types of selection. And definitely need to understand directional selection, disruptive selection, stabilizing selection. Those are terms that you're definitely going to want to know and be able to understand. Now, sickle cell trait. Already looked at sickle cell trait. Absolutely, College Board loves to ask you about sickle cell trait as far as just how it even developed, understanding our protein synthesis, understanding protein structure, understanding then denaturing of proteins. All this is all part of it. But what we also have to understand here is why this particular trait exists is because of heterozygote advantage. So this was a protection against malaria. So if you are a heterozygote, and so you've got one allele for normal hemoglobin and one allele for sickle cell, you're actually resistant to malaria. So this is what is referred to as the heterozygote advantage. So in a heterozygote advantage, would we ever, so in this particular case, you're homozygous recessive for sickle cell, in a third world country without any medical care, without any intervention, that offspring is probably not going to survive. But that's always going to be a possibility because that recessive allele will never go away, because that recessive allele is an advantage if you're now a carrier. Now, I'll also talk about this one here. This is frequency-dependent selection. Now, this is actually pretty cool. So if we actually take a look at these little fish, so there are right-mouthed microlepis. So they actually go to the right. So their mouth is shaped to the right. That's the way it is built. That is their phenotype. These guys up here, left mouth microlepis. So these guys up here, well, they would clean this side of this fish. So in cleaning that side of the fish, that's their food source. But what would happen if now there's lots of these guys reproducing? If lots of left mouth microlepis are reproducing, well, now there's going to be competition for food, there's going to be competition for reproduction, and their population is going to decrease. Thus, then favoring, right mouth polylepis, well, then there's more food for them because nobody's sucking off the right side of this orange fish up here. So now they're going to reproduce. So in this curve, you can actually see this frequency-dependent selection, the frequency meaning what are those characteristics within a population that now can actually kind of have this shift that's happening. Now, Mr. Peacock. When Hardy Weinberg said that mating is random, well, in higher level organisms, it's not always random. There's choice involved. And so when you have choice, what choice ends up leading to is some type of sexual dimorphism. And with sexual dimorphism is actually addressing the fact that I have two different morphs in my genders. I have a male who looks one way and the female who looks another way. And so you can tell that when you have this sexual dimorphism, that there is some type of sexual selection going on within the population. Now, these plumes of a peacock's feathers, their whole job is to attract a mate. And so this female is going to select the male based on the feathers. The larger the plume, the more colorful. That's all a signal to her of the set of genes that this particular male has. So this is a choice. Now, when we actually have this particular type of choice, this is actually what we would refer to as an intersexual selection. So inter means from one to the other. Female is choosing the male that she's going to be mating with. So kind of like you guys hear of inter-district transfers. Somebody who was supposed to go to Lincoln High School comes to Whitney High School, different district. Therefore, it's an inter-district or inter. Now, when we talk about something that's an intra-sexual selection, if it's an intrasexual selection, if you guys have ever seen like videos of these male rams butting heads, well, they're competing for the right to mate with the herd of females. So we get these males that look different than the females because it's now male to male competition for who gets that right, or in some cases, it's female female competition of who gets the right to mate and produce more babies. So that would be an intrasexual selection. So that kind of sums up the chapter 21 concepts that I wanted to go over. But kind of that main end to the chapter that is talked about at the very end is why natural selection cannot fashion the perfect organism. Well, you've got the genes that you've got, and you can modify that basic body plan. Here is a sad, sad fact. Humans will never evolve to have wings. If you did, guess what you'd have to give up? You'd have to give up hands because we are four limbs and it's only what we have in that basic body type that can be modified. So basic constraints, historical constraints, existing variations, and really it's chance. I mean, there is no set pattern or path that evolution is going to take any particular species. So perfect organism, does it exist? Yeah, for every environment, it's the perfect organism for what's there at that time, and that's what natural selection does to populations. So, with that, there's chapter 21 wrapped up for you, and my first screencast lecture, and hopefully one or two of you watch it and it helps you guys out. So, peace. Talk to you guys later. And email me or message me on Schoology, and I'll answer any questions that you guys have. So, talk to you guys later. Adios. Let me figure out how to turn this off. Come on, screencast. How do I get you off? Maybe. I'm trying to figure out how to shut me down. Shut you down, Shelton. Oh, wait, here it goes. There we go. Figuring it all out. I could edit it out, but actually this is more funny watching me just kind of struggle and figure out the technology. So the struggle is real and we're all in this boat together as we all figure out this online learning right now. Thank you, COVID-19, for forcing us to do something we're not used to. See ya. Bye.